Welcome back to Microbial Concepts. So, after a long break, I'm back with food microbiology. So, this is first part, and we will be studying about microorganisms in food. Okay, some examples we will be studying today. So, first, microbiology is the science which includes the study of the occurrence and significance of types of microorganisms which are the beginning and ending of intricate food chain, okay, upon which all life depends. Okay, so a food chain is very important and all life forms depend on this food chain. Okay, so the food we eat is actually rarely sterile. Okay, just remember this point. Why? Because the food we eat, it has major components like carbohydrate, fat, proteins, minerals. Okay, so all these nutrients with water, it serves as a good medium or good source for microbial growth. Okay, so our food, when it comes in contact with air or say comes in contact with soil, for example vegetables or fruits, they or you can say some microbes, they get accumulated on the surface. Okay, so if the food is cooked and then it comes in contact with air, then the microbes from air, they may get settled down. Okay, so it is very rare scenario where food is actually sterile. Okay, our food contains many bacteria or fungi. So because food items, they carry one or more microorganisms, and the number depends on which microorganism gains access and how they grow, or how that particular bacteria or fungi that survives and interacts with the food over time. Okay, so depending on these all factors, the number of microorganisms may vary. So microbes can get access while harvesting, processing, transport, or while handling or cooking, or while storage. Okay, the entry or the access may be through any of these steps or processes. The numerical balance between the various type of microbes will be determined by the property of food, its storage environment. Properties of microbes means the one which is growing. If the bacteria is acidophilic, okay, acid loving, then it may grow in a food which is having high pH. Okay, sorry, low pH means acidic pH. And next is effects of processing. That means while processing, washing, or say heating, or cooling, these all processes, it also, or these processes may also hamper the growth of microbes. Okay, so microbes, they manifest their presence in one of the several ways. So first, if they are present on surface of food and the food is kept still for a long time, then they can start decaying the fruit. Okay, or simply they will start spoilage. Okay, next is foodborne illnesses or food poisoning. If for example bacillus cereus grows well in some rice, okay, after cooking, if the rice is not consumed in say 24 hours, it is kept in refrigerator again, and in third or second or third day it is reheated and consumed, then this is where bacillus cereus, it grows, it produces a toxin, and this causes food poisoning. Okay, so foodborne illnesses is also one example. Then third is microbes, they can transform food properties in a beneficial way. Example, fermentation, okay, alcohol formation or wine formation, etc. The first person to appreciate and understand the presence and role of microorganisms in food was Pasteur. In 1837, he showed that the souring of milk was caused by microorganisms, and in 1860, he used heat for the first time to destroy undesirable microbes in wine and beer, okay, so that they can be stored for long time. The first realization that microorganisms were involved in food production processes was in 1837, when scientists, they discovered the role of yeast in alcoholic fermentation. Okay, so this is how microbes they came into picture, and they started, or microbes got good attention and importance in food production or food industry. So mostly bacteria and fungi are found in food, and in some cases viruses are also found. Okay, so now let's see some factors. Briefly we will cover this topic, some factors which affect microbial growth in food. So the factors can be divided as intrinsic factor, okay, extrinsic factor, implicit factor, and processing factor, right? So you can refer to this chart and you can write a short note. If you get a question like describe factors affecting microbial growth, then you can just draw this chart and you can explain each and every factor separately. For now we will just cover briefly. So first is composition of food, okay, temperature, then how much sugar is present, or we can say what is the pH of food. Depending on that, the microbial growth will differ or it may affect. Next is pH, then presence and availability of water, how much moisture is present. Then third is oxidation reduction potential. So this point it is actually related to cooking. Okay, so oxidation reduction potential is altered by cooking. The next is physical structure, then presence of antimicrobial substances. Then the next is temperature, and last relative humidity. Okay, so moving towards next slide, we will see some examples. A good example of microorganisms uses in food production is the process of fermentation. Okay, a very good example and widely studied. So which results in the production of organic acid, alcohols, and esters. This helps to either preserve the food or to generate distinctive new food products. So this helps means fermentation, it helps to either preserve the food, okay, which means for long time you can store your product, or to generate distinctive new food products. Then next is microorganisms are involved in producing many foods and beverages. Then fermentation produces characteristic flavors, aromas, and consistencies of various foods. Okay, so you can get various products, food products, by doing fermentation. Okay, so fermented milk product is, this is what you can write in your answers. So here lactobacillus or lactococcus or bifidobacterium are examples which bacteria which are used in fermented milk products. Then a variety of foods including Indian idli or dosa or rabri, they in this food items, fermentation is done by using Leuconostoc mesenteroides and S. picalis. Okay, so these are some cultures which are used. Then in case of probiotics, a live food supplement used in yogurt and other fermented milk products. It includes Lactobacillus acidophilus and Bifidobacterium bifidum. So these are examples for probiotics. I had uploaded a video on microbiology behind idli or facts behind idli. Okay, so you can watch that video. It is uploaded on my channel. Okay, so do check it out. Then next is about bread. Okay, so baked goods like bread, rice, because of the presence of yeast as a rising or leavening agent. The most common yeast used in bread making is Saccharomyces cerevisiae. One of the most common is Saccharomyces cerevisiae, the same strain used in bread making. This is used to make L-type beers and is known as a top fermenting yeast as it forms a foam on top of the brew. Bottom fermenting yeast such as Saccharomyces pastorianus are more commonly used to make larger uh, quantities. Okay, they ferment more of the sugar in the mixture than top fermenting yeast, giving a cleaner taste. Okay, so these are some examples. So here is a simplified chart, flow chart, so you can remember examples in this way. So first is aspergillus. So species of aspergillus is used for making soy sauce. Okay, the byproduct is actually the lactic acid, which enhances the flavor. Again, lactobacillus is used for cheese or yogurt production, and the byproduct which actually gives the flavor is lactic acid. Then in case of ethanol and in case of beer, wine and bread, Saccharomyces cerevisiae is used, or any other species of Saccharomyces is used, and the byproducts for beer are ethanol and CO2. Then in case of wine, the byproduct is actually ethanol, which gives the flavor or consistency or this texture and etc. In case of bread, carbon dioxide, which is released by the yeast, is caught by the gluten, and that gives the spongy effect, okay, to the bread. Here is another one. It is actually the same, but if you want to write in stepwise manner, like from glycolysis towards the end product, then you should follow this second chart. Okay, you can get some good marks if you do this. So again, fermentation products which you obtain from Propionibacterium is Swiss cheese. In case of aspergillus, you get soy sauce, or in case of lactobacillus and streptococcus faecalis, you get cheddar cheese and yogurt. In case of Saccharomyces, you can get wine, beer. Then if Clostridium is used, it gives or it produces byproducts like acetone and isopropanol, which are used for production of nail polish remover and rubbing alcohol. In case of Escherichia and Acetobacter, acetic acid is produced, which is used for production of vinegar. Okay, so next example is of wine. The alcohol in wine is formed by the fermentation of sugar in grape juice with carbon dioxide as byproduct. So yeast is naturally present on grape skin, okay, and this alone can be sufficient for the fermentation of sugars to alcohol to occur. Okay, so another example is of fungi, which is used to make or produce mycoproteins. Okay, so Fusarium venenatum is a strain which is efficient in converting inorganic nutrients into proteins. Okay, so this is actually a continuous fermentation. So we know that Fusarium is a fungus, and it reproduces quickly and makes a mass of mycelium. So fungal hyphae are removed in, say, some intervals of time. The hyphae is removed and it is processed, and it is then packed. So this hyphae is good meat substitute, and it is actually high in proteins and low in fats. Okay, so next is Penicillium roqueforti. So it is an organism that is used in blue cheese making process, but now a lab strain is also added, and it is carefully controlled to ensure the cheese matures consistently between batches. Okay, so a lab strain, which means control and can be a modified strain of Penicillium roqueforti is being added with the natural strain. Okay, so that it can be carefully controlled to ensure the cheese matches consistently between the batches. Next example is Aspergillus oryzae is used in production of soy sauce. Then Monascus purpurea, a red color mold, is used to alter color of food. Okay, for example, in case of red wine, this mold is used. Then rice contains Bacillus cereus spores that can reproduce harmful bacteria. So when rice is cooked, then these spores, they survive. But when cooked rice is reheated, these spores, they grow into toxin producing bacteria. Okay, and this may cause food poisoning. Okay, so you can skip this example, but this is actually a food poisoning example, but these spores are already present on the surface of rice grain. Okay, so that's why, as we are studying microorganisms in food, I had mentioned this point. The next is fruits and vegetables harbor microorganisms on the surface. So their uh, type and level can vary with soil condition, types of fertilizers, and water air quality. Molds, yeast, lactic acid bacteria, and bacteria from genera like Pseudomonas, Alcaligens, Micrococcus, Erwinia, Bacillus, Clostridium, and Enterobacter can be expected from these sources. Okay, so yeah, we had done with today's topic, that is microorganisms in food. Okay, I might have skipped some points. If you find that this point should be mentioned or this point should be added, then you can write that point in comment box. Okay, so thank you for watching. Like, share, and subscribe to my channel, and do share these videos with your friends. Do follow me on Instagram and Facebook by the same handle, Microbial Concept. Okay, till then, bye bye and all the best.